All right, welcome back. We're going to start here with a new part by talking about the actual action potential itself. So this is what is going to be the electrical signal that we've been building up to that is actually going to travel down our neuron and specifically down the axon of that neuron. So before we talk about the action potential itself, we have to talk about some different types of nerve transmission. So we have things called resting membrane potentials, and we have something called a graded potential. Now, the resting membrane potential is it's really what it sounds like. It's the potential of the neuron whenever it is resting. So the difference in charge across the membrane when a neuron is unstimulated when it is resting. Usually it's somewhere between negative 40 and negative 90 millivolts. And we'll call this neuron polarized. It is polarized because there is a difference in charge with the inside relative to the outside. So the inside is going to be more negative relative to the outside. Okay, and this will change as we progress through our action potential. Okay, so you can imagine you'd have to have more negatively charged compounds and molecules within the cell if the inside is going to be negative relative to the outside. And that's exactly what we have. And then before we have our actual action potential, we can have things called a graded potential. Now we'll learn that an action potential is sometimes referred to as an all or none event. It either happens or it doesn't. You don't kind of have an action potential. You don't have action potentials of different sizes. An action potential is an action potential. We can contrast that with something called a graded potential. Graded potentials are small changes in the resting membrane potential and can go in a couple of ways. We can have something called a depolarizing potential or a hyperpolarizing graded potential. Depolarizing means that we're becoming less negative or more positive. Hyperpolarizing means we're becoming more negative or less positive. And so you can imagine if we're depolarizing, we're trying to get rid of or minimize the polarity, so we're becoming less negative. We're getting closer to neutral, or closer to zero. If we are hyperpolarizing, we are exaggerating this polarity. We are causing our membrane potential to become more negative. Okay, so the way you do this is you allow, if you're talking about depolarizing, you're going to open up sodium, and we know that sodium is primarily outside of the cell, and so it's going to rush in and make the inside of the cell less negative. This is called depolarization. You could have the hyperpolarization, which is the opposite, involving potassium. In this instance, a stimulus would cause the opening of potassium voltage gated channels. Potassium is highly concentrated inside the cell, so when those channels open, potassium rushes out, taking with it its negative charge, and it causes, taking with it its positive charge, and that makes the inside of the cell more negative. So you exacerbate and exaggerate this polarity. Okay, so these are important, and we'll talk about how you can take different graded potentials and we can summate them and we can kind of add them up until we get to something known as threshold, and we'll talk about threshold. So here you can see an example of depolarization and hyperpolarization. If we are depolarizing, you can note that, okay, so here we're at resting membrane potential. We said somewhere between negative 40 and negative 90, so we're at negative 70. This is resting. When you have some stimulus that causes depolarization, well, what happens to our resting membrane potential? Well, it goes up. It becomes less negative or more positive. And what we're ultimately trying to get to is this dotted line here, this dashed line, which is threshold. And threshold is the minimum voltage that you must achieve in order for an action potential to proceed, to occur. And so if you do not reach threshold, you do not get an action potential, okay? Only if you get threshold. Now, we can contrast this depolarizing stimulus with hyperpolarizing stimulus. Here again, same thing. Here's resting membrane potential. Here is threshold at negative 50. When we get our stimulus, well, boom, now we drop a little bit. We become more negative or less positive, and this results in us moving further away from threshold. So what this means is that it's harder for an action potential to occur now because we have exacerbated and exaggerated our membrane potential. We've gone from negative 70 to say maybe now negative 75 or negative 80. Okay, so we will be able to talk about these in relation to each other and how we can kind of combine these and hopefully get up to this minimum voltage threshold of about negative 50 millivolts. So our action potential, like I said, it is a all or none event. It will either happen or not. There are no differences in sizes of action potentials. Every action potential is the same. What you have to do is you have to get up to your threshold of about negative 50, negative 55 millivolts. So what we have is we have an action potential is only going to occur if we get to threshold, which we've already said is about negative 55 millivolts. So here's our negative 50. We see we're just slightly below that. We're at negative 55 is threshold, and here we're at resting membrane potential, maybe at about negative 70 or so. So we're completely resting at this point, and we'll follow this graph through and see exactly what's happening at each stage of our action potential. So one of the first steps we have is we have voltage gated, after we've reached threshold, after we've reached our negative 55, we have voltage gated sodium channels open. And so what these channels are, there are sodium channels that open and close in response to changes in membrane voltage. So once you reach a certain minimum voltage, these channels will open. And we know that ions tend to move from areas of high concentration to low concentration. And we have sodium in a high concentration outside of the cell. So it's highly concentrated in the extracellular space. So when these ion channels open, sodium rushes down its concentration gradient into the cell, causing the cell to become less negative or more positive. And this process of sodium coming in is known as depolarization. So if we look at this picture here, we have come up, we've hit threshold, and as soon as we hit threshold, what do you see? You see this sharp increase in membrane voltage, and this is due to sodium rapidly rushing into the cell, and we call this depolarization, and we are becoming much less negative. So we're coming up to somewhere around positive 35, maybe 40 or 50 millivolts. Okay. Once you reach a maximum voltage, we'll transition into this next phase called repolarization. So what we have is we have potassium channels, voltage-gated potassium channels now open. So we just depolarized our cell. We took away this polarity and got closer to zero and became more positive. At some point, those sodium channels are going to close. Potassium channels will open. Potassium is highly concentrated inside of the cell, so in the intracellular space, and it moves again from high concentration to low concentration. So when these channels open, these voltage-gated potassium channels open, potassium is going to rush out of the cell, moving down its concentration gradient toward the outside and taking with it its positive charge. So when you take away a positive charge, you're becoming more negative or less positive. So you're reestablishing your polarity. So we're going to call this phase repolarization. So we can see that right here. We're at a maximum threshold. Our sodium channels close, potassium channels open, and you get this dropping membrane potential, or you get this dropping voltage here of your membrane potential back down toward threshold. Okay, so this is due to potassium channels, voltage-gated potassium channels opening, allowing potassium to rush out, moving down its concentration gradient out of the cell, taking with it its negative charge, and beginning to reestablish our membrane potential. But what we have is we have too much potassium open. So if we actually go back here, what you see is this little, this line here, this is our resting membrane potential. We get this small region that actually goes below resting membrane potential. So we became too negative. And what happens is that the voltage-gated potassium channels are slow to respond and slow to close. And so what this allows for is it allows too much potassium to exit the cell. And when too much potassium exits the cell, you get too negative of a membrane potential. So instead of just simply repolarizing, we actually hyperpolarized our cell. We caused the polarity to become greater than what it initially to become greater than what it initially was, which was normally about negative 70. Now we've gone to about negative 71, negative 72 millivolts. Okay, so we've hyperpolarized. We've gone slightly beyond our normal resting membrane potential of -70, right? And so slowly we reestablish this membrane potential. We have the sodium potassium pump reestablish this a little bit. And once we get back to resting membrane potential, we can have the whole process start over again. Okay. Now, you may have noticed on the graph there are two vertical kind of dashed lines indicating two periods, one being a absolute refractory period and one being a relative refractory period. Now, what these are these refer to times at which we cannot generate another action potential. So the absolute refractory period refers to the fact that we absolutely cannot stimulate another action potential. It can't happen. Okay, our sodium channels are completely closed. If they are closed, we cannot we cannot have another action potential proceed because of this fact. Now, our relative refractory period, establishing another and generating another action potential is relative. So what you have is you have our sodium channels closing and our potassium channels opening, and what you have is, or rather what you require is a greater stimulus to then produce the same action potential. Okay, so you can have another action potential in this relative refractory period, but it's going to require a greater stimulus than what we normally had. Okay, and this is because our potassium channels are open, making things more negative. So we're going to have to recruit a larger stimulus for an action potential. But again, the action potential itself doesn't change. The action potential is the same, but the stimulus required to generate the action potential changes. Okay, so here you can see, here's our absolute refractory period. Okay, we've stimulated an action potential. We can't stimulate another one until we get down to about this point where we have those potassium channels opening and our sodium channels closed. We can stimulate another one, but it's going to require a greater stimulus because of this potassium quickly exiting the cell. Okay. So we have different ways that this action potential can be traveling and be propagated along our axon. We have continuous conduction and saltatory conduction. We've briefly mentioned saltatory conduction, but we're going to talk about these two right now. Continuous conduction. Now, based on the name, you would think that a continuous conduction of an action potential would be very quick. It would be fast because it's continuous, it's not being interrupted. But it turns out that continuous action potential, the continuous conduction of an action potential, occurs along unmyelinated axons, and we've said that if an axon is unmyelinated, it is going to propagate an action potential more slowly than a myelinated axon. Okay, and so what you get is you have to have, we talked about this depolarization and repolarization. If this has to happen at every single point along an axon, you can imagine that's going to take some time. It's going to take some time and some energy. And so that's going to slow things down. Whereas with our saltatory conduction of an action potential occurring along myelinated axons, we have this myelin wrapping our axon, and we have these nodes of Ranvier, remember? Now, it's at these nodes of Ranvier where you have the depolarization and repolarization. And so if the depolarization and repolarization only have to occur at these very specific points, then you can imagine how it's going to occur more quickly. And why we call it saltatory is the Latin word saltare means to jump or leap. And we'll see a picture, and it really looks like our action potential is starting at one location, and it's leaping to the next node of Ranvier, and then it's leaping to the next node and the next node. And it's because it's only at those nodes where you can have depolarization and repolarization occur. So this process is very, very fast. Okay. So if we look at this picture, if we look at the top, we'll look at a myelinated axon. So this is going to be saltatory conduction. We have our action potential be originated at the axon hillock because that's the only location it can originate. So you would have depolarization and repolarization occur here. And then you would have it jump and bounce to this first node. Depolarization, repolarization occurs, and then you have it occur here. It jumps to here, same thing happens, and it jumps to the next one as well. Whereas if we are talking about an unmyelinated axon or continuous conduction of an action potential, note that you have to have this whole process occur at every single point along the axon. So this is going to be much more time consuming than doing it this way with this saltatory conduction. Okay, so hopefully this makes sense. You can see that we become negative on the inside, positive on the outside, and it changes. So we have sodium rush in, we become more positive, and this spreads to the next area where it continues to occur all the way down our axon of our neuron. So, what is happening in multiple sclerosis? Have you heard it? Maybe you've heard of multiple sclerosis. Sometimes it's referred to as just MS. Well, in MS, I'll go back. In MS, it is a demyelinating disease. Okay, so if you take axons that are normally myelinated, and this can be in the brain, if you take axons that are normally myelinated and they undergo this saltatory impulse conduction of an action potential, very fast, very efficient, and now you take that myelin away, you've lost this ability for action potentials to be propagated very quickly. And so what you see in multiple sclerosis is you may see fatigue, you may see people be very tired, very weak, and the reason for this is their nerves are essentially not functioning properly. Their impulses are being delayed and being carried very slowly compared to what they should be along myelinated axons. Okay, so it's a demyelinating disease that slows the impulse conduction of action potentials, and this manifests in several different ways in the individual. So we will stop here and we'll pick up with synapses in the next part.